So we're ready to start the next session of the workshop. The next speaker will be Sarkis Mazmanian. Sarkis is the Lewis and Nellie Sue Professor of Microbiology in the Division of Biology and Biological Engineering at Caltech, and is also co-host of the workshop. Sarkis will be giving a presentation titled Gut-Brain Connections to Behaviors in Mice. Sarkis.
Thank you very much, Steve. And I hope you can see my slides. And so today I'll talk about some new work leveraging previous research from the laboratory on how microbial metabolites affect behaviors in mouse models. And so, of course, Rob and Sergio introduced some of the concepts in terms of the background on the microbiome, but essentially we have 100 trillion bacteria, mostly living in our gut, that outnumber our own cells, and importantly, the genomes of these bacteria, known as the microbiome, outnumber our own genes by 100 to 100-fold. So these organisms are contributing immensely to our biology. And I think we're, as I mentioned earlier, just scratching the surface of our understanding of the microbiome. Related to various talks, and in particular this one, the microbiome has the metabolic capacity of the liver, so relating it to human organs, and in fact has an equivalent mass as the human brain. And largely in mouse models, the microbiome has been implicated in a number of disorders and neurological conditions, including autism, which is what I'll speak about today, but also anxiety, depression, Alzheimer's, and Parkinson's disease. Lots of research linking the microbiome to metabolic disorders, including cardiovascular disease, a lot of work in obesity and type 2 diabetes, and immunologic disorders, as we've already heard from Sergio, in terms of multiple sclerosis, but also inflammatory bowel disease, arthritis, and a variety of different infections. And so the microbiome looks like it has lots of effects on our biology, but again, zeroing in on one particular behavioral disorder, namely autism. Autism affects about 3 million people in the world, largely children, and in the US, about 1 million individuals and their families are affected by this disorder. Current estimates are that about 1 in 54 births in the US result in a diagnosis of autism, making this one of the fastest growing developmental disorders. And you can see on the right the statistics from the CDC, how over the past few years, the prevalence of autism has increased. And so there's still a lot of debate as to what are the factors that mediate this increase. Clearly, awareness and in some cases changes in diagnostic criteria are relevant, but there's also growing evidence and interest in the role of the environment that may help explain this increase. Autism has a tremendous cost, not just societal and personal, but economic as well. And what really got us interested in this topic many years ago in collaboration with Paul Patterson at Caltech is the fact that many children with autism have reported to have gastrointestinal issues that range from abdominal cramps, bloating, constipation, and diarrhea. And a wealth of studies, some of which you'll hear about from Rosie later today, on how the microbiome is different between individuals with autism and healthy controls. And so autism is a behavioral disorder. It is diagnosed based on instruments or tests that compare behaviors between individuals in the population. And the 3 major domains within which autism is diagnosed are the behavioral domains that involve social and verbal communication, as well as repetitive behaviors. However, many individuals with autism show really a wide variety of whole body phenotypes that go beyond just behavior, and many of those are listed here. Of course, these are in a subset of individuals with autism. These are not diagnostic criteria, but again, what really caught our eye and suggested that perhaps the microbiome may be involved in autism spectrum disorder are these gastrointestinal symptoms I mentioned, but also a number of reports showing immune dysfunction and metabolic abnormalities. And again, as I just mentioned, those are systems in the body that appear to be regulated or impacted by the microbiome. And so we started this work with Paul many years ago to ask, in a particular mouse model of autism that he had developed at Caltech, are there gastrointestinal issues? And so I'll just briefly, in the interest of time, introduce the mouse model. There's a lot of work done on this and many other models as well. But Paul leveraged epidemiologic data that show that mothers during pregnancy that experience an infection, a severe infection, usually coupled with a fever, are at increased risk of having a child with an autism diagnosis. And Paul modeled this in animals by essentially giving pregnant females the flu. And so initially he did use live influenza, but ultimately used poly-IC, which is a viral mimic, which activates the immune system, similar to how flu or other viruses activate the immune system, again, in mothers, and then tested the offspring for a variety of different outcomes and showed in a series of studies, not just from Paul's lab, but from now many other laboratories as well, that these animals have a number of behavioral abnormalities associated with autism that are shown here on the left, as well as, really interestingly, neuropathologies, particular effects on migration of cells and altered neurotransmitter signaling in these animals that is reminiscent of some cases of autism. And so leveraging this mouse model, the work that we did with Paul, which was in collaboration with Lane Tsao, published in 2013, was to show that in this animal model of autism-like behaviors, that the mice had gastrointestinal symptoms. I won't show the data here, but they also had an altered microbiome, which was implicated as well. And what we showed is that these animals have a phenotype called leaky gut, which essentially is an effect on barrier permeability. And so this assay measures how much of a fluorescent dye leaks across the epithelium. And you can see in these offspring of poly-IC treated animals in the white bars that there's more leakiness in an animal that has behavioral deficits compared to its control. And then we surmised that a probiotic that we had been studying in our laboratory that has anti-inflammatory effects, that's therapeutic in mouse models of intestinal inflammation, may be helpful in the gastrointestinal symptoms associated in this mouse model. And so we treated these animals with a human commensal called Bacteroides fragilis, and this is just an oral treatment to these animals, and showed that Bacteroides fragilis treatment leads to a correction of this phenotype of leakiness across the intestinal epithelium. And this just shows that the leakiness was there prior to the treatment of with the probiotic. And so the cellular and molecular basis for this effect is mediated by the expression of tight junction protein. So tight junction proteins essentially hold our intestinal epithelial cells together, forming the barrier between the gut and our bodies. And we showed by RNA and by protein that there are differences in various tight junction proteins, namely claudin 8, which decreases in the offspring of poly-IC treated animals and then is then restored by Bacteroides fragilis treatment. And then this claudin 15 phenotype, where claudin 15 is increased and then also restored by Bacteroides fragilis treatment. This pattern has been shown in other mouse models to be associated with leaky gut. And then here we're just staining for claudin 8, showing that Bacteroides fragilis actually induces an increase in claudin 8 expression across the epithelium. So this is an intestinal epithelial cell, and you can see the very bright red staining is the expression of claudin 8. And so concomitant with this improvement in the gastrointestinal symptoms, we also showed that Bacteroides fragilis improves behaviors in MIA offspring. So here we're looking at vocalization behavior as measured by ultrasonic vocalization, and we can look at a number of different parameters in this test. Here we're just looking at the number of calls, and you can see that poly-IC offspring call less than their wild-type counterparts, and the duration of calls and poly-IC offspring, when they do emit a call, the duration of that call is shorter. And in both cases, treatment with Bactris fragilis increased both the number of calls as well as the duration of calls, suggesting that it is improving this core symptom of autism as can be modeled in mice. As I mentioned, autism is marked by repetitive behavior phenotypes as well. And one test to look at repetitive or stereotype behaviors is to perform a marble bearing test, where we look at the number of marbles buried by a mouse in a given amount of time. And again, the offspring of poly-IC treated animals bury more marbles than that of controls. And when these poly-IC offspring themselves are treated with Bactris fragilis, you can see that the number of marbles buried is decreased back to wild-type levels, again, suggesting that there's a restoration of this core feature of autism. And there were a number of studies that we did in that original paper, but one that led to further research was to try and understand what is this gut-brain connection, or one aspect of the gut-brain connection in this mouse model of neurodevelopment. And we looked at circulating metabolites, small molecules, in the various groups of mice and asked, are there differences in particular molecules or metabolites? And there were a number of differences that we reported, but one that caught our eye is this elevation of a metabolite called 4-ethylphenyl sulfate, which I'll abbreviate with by just calling it 4-EPS. 4-ethylphenyl sulfate is increased in the poly-IC treated animals in the MIA model, and then reduced by Bactris fragilis treatment. And so this was over a 40-fold increase. In fact, in our data set, this metabolite was the most highly dysregulated of all the metabolites that we looked at. And so what is 4-EPS? It's a tyrosine metabolite that's converted into 4-ethylphenyl, and then the host, likely in the liver, sulfates 4-ethylphenyl into 4-EPS. And I also want to draw your attention to the fact that 4-EPS is one metabolite. Again, it was the most highly dysregulated, but there were many metabolites that we identified to be altered in this particular mouse model. And here's a handful of those. And they vary in different chemical classes, but many of them seem to be products of tyrosine and tryptophan precursors. And so it appears that 4-EPS is of microbial origin. When we looked in just wild-type animals, comparing germ-free animals to SPF mice, one can see that germ-free animals have very low to undetectable levels of 4-EPS compared to SPF mice, suggesting, not conclusive, but suggesting that 4-EPS is of microbial origin. And looking at non-MIA models, we showed in the catnap2 animal that there's an increase in serum 4-EPS in this other genetic mouse model of autism. And so recently we, in collaboration with Paul Ashwood at UC Davis and Alessio Fasano at MGH, were able to collect plasma and fecal samples from a large cohort studied at UC Davis. This is the CHARGE cohort. And we did untargeted metabolomics on both tissues, on both plasma and feces. And I'll just zero in again. We found many, many changes in this paper, which is on bioRxiv if anyone's interested. And just zeroing on 4-ethylphenyl sulfate, there is in now a human cohort this increase in 4-ethylphenyl sulfate in a handful of individuals with autism. So 129 samples or people with autism, 129 plasma samples, 100 typically developing samples, so quite a large cohort. And again, while 4-EPS is found in the general population, there seems to be this elevation in individuals. And if one were to look at the fold elevation, there's actually a little bit of a difference between individuals who had GI symptoms and didn't have GI symptoms. But not surprisingly, individuals with an autism diagnosis that also have gastrointestinal symptoms have the highest elevation of 4-EPS relative to typically developing individuals, almost a 9-fold increase. So we focus on the biology of this one metabolite, again, as a surrogate for many other metabolites that may have biological effects. And so the data I showed you suggests that maybe 4-EPS and related metabolites are biomarkers, but could they also be bioactive? And so we started working with Michael Fischbach a few years ago to set up mouse models to directly test the function of 4-EPS, and doing a disservice to Michael and his postdoc Masa for what is a tremendous amount of work summarized in one slide, where they essentially put together the entire biochemical pathway. A lot of it was discovered in novel chemistries in the microbiome, and generated strains of bacteria that were engineered to convert tyrosine through these two metabolites to 4-ethylphenol, so engineered Bacteroides ovata strain and the Lactobacillus plantarum strain to now produce 4-ethylphenol from tyrosine, and then again the host sulfates for ethylphenol, largely in the liver, but may actually happen in the colon as well. So now we have two sets of bacteria that are identical in every way other than the fact that one group expresses 4-ethylphenol and the other bacteria do not express 4-ethylphenol. And so Brittany Needham in our laboratory colonized two groups of mice with these bacteria. And so these are different groups of mice, genetically identical in every way, except that they either harbor bacteria that do not produce 4-ethylphenol or do produce 4-ethylphenol, and as measured in the feces. But then when she looked in the urine, you can see this increase in 4-ethylphenol sulfate. And quite intriguingly, if we block the efflux of molecules out of the brain, we can now detect 4-ethylphenol sulfate in the brains of these mice as well. Again, 4-ethylphenol is produced in the gut, and 4-ethylphenol sulfate is detected in the brain. And then in collaboration with Daniel Holschneider at USC, we performed 2-deoxyglucose staining, essentially looking at activity in different brain regions of animals. And these are the results of the autoradiograms, looking at the elevation of activity in animals, overlaying 4-EP above 4-EP positive above 4-EP negative. And the regions in red are those that are activated. And you can see the hypothalamus, the amygdala, the BNST, and the paraventricular nucleus of the thalamus are all regions that are activated. And interestingly, there are some regions that are reduced in activation in the 4-EP positive group. And there's also data showing that there are connectivity differences in essentially different regions of the brain that are activated or not communicating or connected to each other in the 4-EP positive animals as they would be in the 4-EP negative or otherwise control animals. And so showing brain activity, and especially I'll focus on the PVT, was inspired us to now look at a cellular molecular level of what's happening in the brain. And indeed, the largest number of gene changes, and this is an RNA-seq experiment of different brain regions in these animals, the largest number of genes that were dysregulated or altered are in the PVT, and you can see that here. Neurons are certainly affected, but what was really interesting and potentially striking is that there appeared to be an effect on oligodendrocytes, and I'll explain what oligodendrocytes are in a minute. But this initial gene expression profile suggested that there is an arrest or a decrease in the maturation of oligodendrocytes. And one can now look at individual genes associated with oligodendrocyte precursors or mature oligodendrocytes. And looking at all of these genes across various different pathways, one can see that the genes associated with oligodendrocyte precursors are elevated in the 4-EP positive animals. Those are in the dark green. And then the genes that are associated with mature oligodendrocytes are reduced. And you can see all of these genes that are patterns of mature oligodendrocytes are reduced in the animals exposed to 4-EP. And so this again predicted an effect on oligodendrocyte maturation. And oligodendrocytes are cells in our brains that myelinate neurons. So their differentiation, their development is highly regulated. And only when these oligodendrocyte precursors become mature oligodendrocytes do they begin to myelinate our neurons, and myelination of neurons are important for the activity of neuronal cells. And so we wanted to test this prediction. And so Brittany looked at the differentiation or developmental state of oligodendrocytes in the brains and showed that, looking at a maturity coefficient, that there's a decrease in mature oligodendrocytes in the brains of the 4EP exposed animals compared to the 4EP negative. There's no difference in total oligodendrocytes, so we believe that it's not that the cells don't exist, but they're just not maturing into mature oligodendrocytes. And this was done by imaging, but she backed this up with flow cytometry data, which essentially shows the same thing, that looking on a cellular basis, that there's a decrease in mature oligodendrocytes in mice that were exposed to 4EP relative to the 4EP negative group. And so as I mentioned, oligodendrocytes myelinate neurons, and so we looked at myelination, and I'll just go quickly through this data for the sake of time, is that we showed that there are more unmyelinated oligodendrocytes in the groups of animals exposed to 4EP. The G ratio looks at myelin irrespective of the cell shape, and a higher G ratio is a sign of lower myelination. You can see that there's an increase in this G ratio for the 4EP positive group. And then when we just looked at myelination thickness, and this was work done by Mark Ladinsky and Pamela Bjorkman's lab here at Caltech, that the thickness of the myelin is reduced in the 4EP positive group. And so ultimately, this translated into behavioral effects across a variety of different domains. We looked at the open field test, which measures anxiety in an animal. The more often an animal enters this square in the middle of the arena, the less anxious it is. And so you can see that anxiety is increased in the 4EP positive group because these animals enter the center less. They're less bold than the 4EP negative group. There's no effect on locomotion, so it's not as if the mice are just lethargic. We've looked at many other parameters of physiology in these animals. There's no sign of illness in these animals. It appears that they're choosing to enter the center less. We've also looked at elevated plus maze, where the bolder animals will go on these open arms, whereas the animals that have an increased anxiety feature will stay in the arms that are closed. And you can see that there are less animals entering these open termini if the animals were exposed to 4EP in the gut. Specifically for autism-like behaviors, we see a change in social interaction, a decrease overall in total social interaction. It doesn't reach significance, but this interesting increase in a specific activity of anogenital sniffing. And then going back to the USV test, there's less vocalization in these animals that have 4EPS in their brains. And then finally, to demonstrate that this effect is through oligodendrocytes, we use a chemical called clemistine fumarate, which artificially drives the maturation of oligodendrocytes. So even in the presence of 4EP, we can force the maturation of oligodendrocytes, and you can see that here. So this immature phenotype that I already showed you in the 4EP positive group, upon treatment with clemistine fumarate, results in a maturing of these oligodendrocytes. And that also then results in an improvement in behavior. So you can see in the elevated plus maze that these timid animals are no longer shy. They enter into the open arm as similarly as the 4EP negative group. When we looked at the open field test, again, you can see a restoration of this anxiety-like phenotype. And as I mentioned, marble burying, this repetitive behavior is also a core feature of autism that is increased in the animals that are exposed to 4EP, but then maturing of their oligodendrocytes with clemistine fumarate results in a more normal number of marbles buried. And so just to summarize the data that I've shown you for one of these metabolites is that coupled with the changes that we know exist in the ASD microbiome, perhaps there are changes in the metabolome in the gut as well, concomitant to those changes. It's very hard to know what changes the microbiome, but clearly I think we know that there can be changes in both molecules and metabolites. There's an increase in the flux of these micrometabolites across the intestinal barrier. 4-ethylphenyl is sulfated to 4-EPS. Those concentrations increase in the circulation. And then ultimately, 4-EPS enters the brain. Though we don't know if 4-EPS in the brain is what mediates this effect. We just know that it's there. But ultimately, resulting in the rest of oligodendrocyte maturation, reduced myelination, and these altered behaviors. I'll stop by thanking Brittany, who did the work, and again, sorry for being a little bit over. These are all of our collaborators I've mentioned, and again, all this work was inspired by the late Paul Patterson. Thank you very much.
Thank you, Sarkis. That was very exciting. A couple of questions. I'll read one from the audience. It seems that 4-APS in female C57 mice is much higher. How do you explain this, especially in the context of males being more affected by autism spectrum disorder? How does the 2DG data break down across the sexes?
So regarding the first question, so those were data in just black 6 mice. So that's not a mouse model of autism. And the dimorphism in 4-APS is one that we can't explain. We don't necessarily see a difference between male and female mice in this mouse model of behavior. In fact, there are about 40 mouse models of behavior that I know of that span genetic, spontaneous, or even induced modes and etiologies. And only one of them, again, to my knowledge, shows this gender bias, but it is true that there's about a 4 or 4.5 to 1 ratio of diagnosis of males to females. So autism is in the human population is much more prominent in males than females, but largely the mouse models don't capture that that gender bias. And in terms of the 2DG gel, I can't recall off the top of my head if there's a gender bias. There may be, and I can get back in the and answer that in the Q&A. But again, there's no gender bias in the behaviors, which are the more relevant readout for autism.
Thank you. Another question. Do you think radiation-induced changes in gut leakiness will play a significant role?
It might. And so radiation, whether it's chemotherapy, whether it's ahead of other procedures or in space, is known to affect stem cells in the gut, which then impacts the epithelial barrier because epithelial cells differentiate from gut-derived stem cells. And so there's growing evidence that radiation and other treatments such as chemotherapy that affect stem cells will cause leaky gut, will cause barrier defects. Will that result in autism? I doubt it. I wouldn't be surprised if it would result in changes in behavior, or maybe part of the physiological process for changes in behavior. And I say that because this leaky gut phenotype, it's been shown in a couple of cohorts of autism, but it's also been demonstrated in other neurodevelopmental and even neurodegenerative diseases. So leaky gut has been measured in schizophrenia and in depression. It's also been measured in Parkinson's disease. And so there may be a more broad effect than just autism or even emotional behaviors for the effects of leaky gut. And that's certainly, more on the sort of the neurological side. I wouldn't be surprised if there are widespread metabolic changes as well due to leaky gut. And in fact, this is an area that I know certain drug companies are quite interested in. There are assets in the clinic as we speak targeted to improving leaky gut.
Great, thank you. Do you have any sense of what the biochemical mechanisms are for APS acting on various cells?
We don't know what the cell type is that senses or responds to 4-EPS. We don't know what the receptor is for this molecule. I mean, it sort of falls into a class of neurotransmitters, like many other neurotransmitters like dopamine, which are tyrosine metabolites. And I also want to mention that the target of 4-EPS may be outside of the brain. So we don't know if there's a direct effect on oligodendrocytes or on neurons, that there may be some relay between a peripheral target, a target outside of the brain that may sense 4-EPS, and then transmit a signal into the brain. We have some very preliminary in vitro culture data that suggests that 4-EPS may affect maturation of oligodendrocytes or may affect the differentiation of neurons. But again, those are very, very preliminary experiments. And we would love, and that's one of the next phases of this project, is to identify the target of 4-APS.
And the final question, a more general question from the audience. Does the microbiome terminology represent bacteria in the gut, intestinal tissue only? Is there evidence showing the microbiome in other tissues, for example, brain, lung, bone, marrow, etc.? If there is such evidence, are they similar or different from those found in the gut?
So Rob alluded to this. So the microbiome in humans is largely found in the gut, but many other environmentally exposed surfaces, almost all environmentally exposed surfaces of the body are colonized by bacteria. They're quite different from each other in terms of our own biogeography. And so maybe one way of impressing upon the audience the point of how different the geographies are is that, and I'll just use you and me as an example, Steve, you and I, our gut microbiomes are more similar to each other than my gut microbiome is to my skin microbiome. And so, the context and the environment within which a microbiome is found is critical to what organisms live there.
Great. Thank you, Sarkis.